Okay. I've been pollinated. It's driving me crazy. My eyes are running, and if I sniff at you, I'm sorry. But I will, in a sense, here. We've been talking about RNA, and we'll continue talking about RNA processing, and we're going to continue talking about how it's done in the life. It's done primarily in eukaryotic cells. Pretty much there's no processing in prokaryotes except for tRNAs, but certainly in messenger RNAs, intervening sequences, done in eukaryotic cells. There's actually a gradation of it. There are less genes in yeast that have intervening sequences. There are more Saussura, and they're the most in mammals. In mammals, almost all of the messenger RNAs are expressed in intervening sequences. When we talk about transcription, we talk about the gene, the coding region, promoter, and part of the gene that codes for protein. Primary transcript, the mRNA, they call it an mRNA transcript. It is the original transcript, but it's the primary transcript. It's also called heterogeneous RNA. The reason is when people who were first working with RNAs, there was tRNA, and there were 20 of those, and ribosomal RNA, there were only 4 of those. And then there was this whole group of RNAs that varied in size, and they were very heterogeneous. There were like 10,000 of them in a cell, or a couple thousand anyway. And so they're called heterogeneous. They are essentially linear with the gene. Everything that's coded in the gene is transcribed into a piece of RNA. That's the primary transcript. It also has the intervening sequences, these sequences that interrupt exons around the gene. And as those primary transcripts are processed inside the nucleus of the eukaryotic cell, and they have to be processed, there are controls that say they have to be processed before they get out of the nucleus. 3 things happen. They have a 5'7" G capsid on the end. It ought to be up here also. They have a poly-A tail put on the end. It ought to be down here also. It's done in these things here are the same. And then that is moved out into the nucleus after all 3 of those events. Removal of introns, 5' capping, and poly-A tail capsid. Then it's moved out into the cytosol of the cell for translation by ribosome. So the question is, how are these intervening sequences moved out? They vary in number. Remember I said the primary transcripts are about 10 times larger than the final, on average, than the final messenger RNAs. They can have differing numbers of introns and exons. The record is well over 50. It's about 60 now. There's a gene that has 60 introns. Crazy. Some of these can be long. They can average, or I don't know what the average, but some of them can be 50,000 nucleotides long. The coding regions themselves, the exons, average about 100 to 250 nucleotides or so. And those that have the most intervening sequences, that have the longest primary transcript, are genes that encode proteins that are found in the extracellular matrix, particularly collagen and fibronectin and things like that. Some of the genes that are involved in moving ions in and out of cells, the cystic fibrosis gene has about 50 introns. The gene that's involved in muscular dystrophy has about 50 introns. So there's a lot of these, and they have to all be processed out. In order to process out, there has to be a common mechanism. We said there's a common mechanism to remove additional introns, and that mechanism is based on 3 consensus sequences. One that occurs at the 5' end of the exon, or the intron, and at the very 5' end, there's 2 nucleotides that are perfectly conserved in about 95% of all introns that are processed. Maybe even more, probably 97% of all introns have exactly the same sequence. The other 3% are processed in slightly different ways, but very similar process. They just evolved, we don't know why, but they've evolved slightly different regions that different proteins deal with those. But on most of them, there's a GU that's conserved at the 5' end. The consensus sequence at the 3' end also has, at the very 3' end, has 2 nucleotides and a G that are perfectly conserved. So there's conservation at the ends of the introns, which are involved, which are necessary to recognize that's where the intron is. There's also a consensus sequence, not in the middle as this sort of implies, but closer to the 3' end, approximately 30 to 50 nucleotides in here perhaps. And that's what we call branch point A. There's an adenosine residue that's absolutely always conserved. It's absolutely essential for the formation of the lariat that we talked about in order to cut out These three recognition sites are used to recognize the intron, and it results in the intron being cut out. And then the two pieces of RNA, exon 1 and exon 2, that end in a G, always G's, have to be ligated together, okay, to form, and then we have two exons together. That's necessary because ribosomes, when they translate, they get hop on the end of the message, and they read triplet codes that go through. If the intron is there, they would just read those triplets, although the introns are cut out, they're not used for translation, so the introns evolve very quickly. And usually within an intron, somewhere there's a stop sequence, an AUG or a GUA, not AUG, but UGA or a UAA or so forth. One of the stop sequences in there, and it ends up in a nonsense protein that doesn't code for a functional protein. Okay, so how are these guys cut out? How are those recognition sequences used? Essentially, they're used by the formation of what we call a lariat. And the lariat forms when there's a cut in the 5' end of the mRNA, the precursor mRNA, and that free hydroxyl, the free 2' the free phosphate that results in the cut, that phosphate right there in the diagram, is used to phosphodiester bond with the 2' hydroxyl of that branch point A. So this is the branch point A. It has the 3' hydroxyl and it's in a phosphate bond with that tyrosine. The 2' hydroxyl is free. It interacts with the phosphate at this branch point A there and forms a phosphoester bond. That adenosine now has 3 phosphoester bonds. When we see that, I know that it does. Okay? When this end of this intron, this end of the intron here, let me see how this. So this is exon 1 right here. When that exon is released, right, the phosphate is there. It forms a lariat. That's the phosphate of this phosphoester. And that guanosine residue now has a hydroxyl, a free hydroxyl, a free 3' hydroxyl. And that hydroxyl can undertake a nucleophilic attack on this phosphoester bond here at the 3' end of the intron, or the 5' end of exon 2. In doing so, it forms a phosphoester bond, and that phosphoester bond then links exon 1 and exon 2. So here's exon 1 and exon 2. They're linked together now. As this does that, it frees up the lariat, right? The lariat now has a 3' hydroxyl, and this is the intron that has been cut out. Everybody see that? Now, this process is essentially requires no energy. This just shows you the same reaction here. That hydroxyl that is going to the nucleophilic attack on this thing. Here's the RNA, right? And here's the phosphate. So this is one exon. This would be the intron, and this is where it loops around there, and you get that phospholytic attack. And what does it show? It doesn't show much more. The 2' hydroxyl. It's the 2' hydroxyl undergoes that, takes that phospholytic attack. This is just another cartoon that's from another textbook. Shows you exactly the same thing. Thought I had another slide that I can tell you about. Maybe I should tell you first. I can tell you. So this is another cartoon. It's just from another text. Shows exactly the same thing. A couple interesting points. If you notice that in this picture here, in this cartoon, that branch point A, which is closer to the 3' end, is actually sticking out. It's almost like an umbilical sticking out there. And I'll show you later when we talk about the mechanism that that's in fact true. That A is pushed out in that RNA molecule. The rest of this is in hybrid with another RNA molecule. I'll show you that. But that branch point A is actually pushed out and it goes there. In order to have this hydrolytic attack, all that has to happen is that A has to be pushed very close to this phosphoester bond. Not this one, not this one, not this one, but that one, right? And that's what this whole splicing process or processing mechanism does, is that it rearranges this RNA, this primary RNA transcript, so that the proper nucleotides, right, the branch point A, the one right at the beginning of the intron, are very close together. And when they do, there's that hydrolytic attack there. And we form there's that adenosine. Now, the other thing that I wanted to tell you, I thought I had a slide, and I'll explain it to you here, is that you notice in this reaction, there's no ATP there. It's energy independent. There's no ATP required. The reason is that this reaction here is what we call a transesterification. Okay? A sterification, the formation of an ester, a phosphoester. A transesterification means going from one ester to another ester. So essentially what's happening is when this biochemistry occurs here, the reaction occurs, the cleavage of this phosphoester bond, right, between the intron and the exon. So that that 3' hydroxyl 5' phospho bond, the cleavage of that bond releases energy. And the release of that energy is used to form the phosphoester bond between that 2' hydroxyl and the free phosphate that's released. So it's a transesterification because we're going from this ester here to this ester here, right? This is going to pull over like this and form that ester there. The energy of this ester bond is used to form this ester bond. Transesterification. No energy required, right? Same thing here. Once this is free now, this is free, right? This hydroxyl can undertake a nucleophilic attack on the 5' or the 5' end of the next exon, or the 3' end of the intron there. That hydrolytic attack cleaves that thing there. And the reaction that puts this exon together with this exon 1 with exon 2, like this, right there, is a transesterification reaction, the second transesterification. The energy of this ester linkage is transferred to the energy of this ester linkage. So there's two ATP-independent transesterifications that actually lead or lead to the formation of the new bonds in the malaria and in the two exons that are put together. Does everybody see that? Does any have a question? No. Yes?
What happens to the lariat after?
The lariats are actually totally turned over. They're degraded in the nucleus by nuclease, which is rather strange, right? Because some of these are huge, 50,000 nucleotides. So the cell has used, even though this is energy independent, that little reaction there, the energy is used a huge amount of energy to put together 50,000 nucleotides on an intron that is going to do nothing more than cleave. Right? So why does it do that? That's the question. Anybody else? Okay. So what directs this? I said that we have these consensus sequences that essentially define, or partially define, and I'll show you what I mean by partially later, but partially define the cleavage sites. And we also said that all you have to do is put these things together in order for this hydrolytic attack and then the transesterification to occur. The question is, how do you put them together and what's involved in this mechanism? And the answer is that there are little molecules